Welcome to Biomindscape. Today we are going to know another topic of immunology, that is dendritic cell. They are specialized antigen presenting cell that form a critical bridge between the innate and adaptive immunity. First, they were identified by a Nobel Prize winner of 2011, that is Steinman. They are highly efficient at antigen uptake, these dendritic cell, and processing and presenting. So they originate from various hematopoietic stem cell, which further getting converted into myeloid or lymphoid progenitor in the lineage. Then they are located in the skin like Langerhans cell and mucosa and peripheral tissues. The immature dendritic cell, they are present on the skin, mucosa and peripheral tissues, whereas the mature dendritic cell, they migrate to the secondary lymphoid organ like lymph node, and the growth factor that are involved are CSF, that is growth GM CSF, interleukin 4, FLT3L. It stimulates the DC development. So mature dendritic cells have been able to present antigen in a lymphoid tissue and to prime active and expand the immune effector cell with unique feature and cytokine profile, whereas the immature dendritic cell, they are not activated dendritic cell and are usually located in the peripheral non-lymphoid tissues, and they can present self antigen to T cells that lead to the immune tolerance either through the T cell deletions or through differentiations of the regulatory T cells. Then myeloid dendritic cells, they are conventional dendritic cells. They are derived from the myeloid progenitor cell in the bone marrow and are characterized by the expression of CD11C that can be subdivided into three groups like monocyte derived dendritic cells, CD1A intestinal dendritic cell, and CD1A Langerhans cells. Then plasmacytoid dendritic cells, they are differentiated from the lymphoid progenitor cell in the lymphoid tissue. They express the CD123 and product high level of type 1 interferon, and they are also contributing in the inflammatory response in the steady state. In pathogen during inflammatory response, inflammatory dendritic cell are generated from the monocyte. Now let's know the types of dendritic cells. There are various types like conventional dendritic cell, plasmacytoid dendritic cell, Langerhans cells, and monocyte derived dendritic cells. So they are located on the tissues, lymph node, then blood, lymphoid tissues, epidermal, and inflammatory site. Now there can be and match the pairs like which type of tissues are present on which location. Like conventional dendritic cells, they are present on the tissues and lymphoid nodes, and plasmacytoid dendritic cell, they are present on the blood or lymphoid tissues, and Langerhans cell, they are present in the epidermis, and monocytic derived cells, they are present in the inflammatory site, and they have various role. Like the conventional dendritic cells, their roles is best at antigen presentations to native T cells, whereas cytoid, these dendritic cells, which are present on the blood and lymphoid tissues, the roles in the production of type 1 interferon, that is INF alpha and beta during the viral infection, whereas the Langerhans cell, they are present on the located on the epidermis, and they reside on the skin dendritic cells, whereas the monocyte derived dendritic cell, they are present on the inflammatory site and they arise during the inflammation or infections. Then let's know about the life cycle of dendritic cell. The immature dendritic cell, they are located in the peripheral tissues. Let's know about the dendritic cell life cycle. First, the bone marrow. It is the origin. Hematopoietic stem cell, that is HCS, give rise to common dendritic cell progenitors, that is CDPs, and it is differentiated into conventional dendritic cells, that is CDC1 and CDC2, and plasmacytoidal dendritic cell, that is PDCS. So monocytes can also be monocyte derived dendritic cell during inflammation. Then immune dendritic cell is peripheral tissues. They leave the bone marrow, they circulate in the blood and enter the tissue like skin, mucus, and intestinal tissue. The functions at this stage is antigenal capture by phagocytosis, macropinocytosis, and endocytosis. So express low MHC and co-stimulatory molecule, not yet good T activators. Example, Langerhans cells in the skin. Then activation is triggered by the pathogen molecule PAMs detecting by TLR an inflammatory cytokine, example TNF alpha and interleukin 1. It changes the occurrence, stop phagocytosis, increase MHC1 and MHC2, CD80, CD86, CD40 and begin producing the cytokine like interleukin 12 for the T helper 1 cell differentiation. Then migration to the lymphoid organ, activated dendritic cell move to the lymphoid vessel, then to the lymph node, that is spleen, and migration guide by the CCR7 receptor on the dendritic cell binding to the chemokine CCL19 and CCL21. Then the mature dendritic cell in lymphoid organ, now fully mature dendritic cell present the antigen on MHC1. They will pass on to the CD8 anti cells, then presenting antigen on MHC2 will pass on to the CD4 T cells. Then they provide the co-stimulator, that is CD80 and 86 with CD28, CD40 and with the CD40L. So this activates the native T cells starting in the adaptive immune responses. So after antigen presentation, the dendritic cell undergo apoptosis. The lifespan is about 2 to 3 days once mature. Prevention overactivation of T cell, avoid autoimmunity. Thanks for watching. Do like, share and subscribe.